Hello people, welcome back to the another lectures on genetics. Today we are going to discuss about structural aberrations of chromosome. This is a type of chromosomal aberration. Just like numerical aberration, it is the structural aberration where the aberration is seen in the structure of chromosome. So let's proceed and discuss. What is chromosomal aberration? Variation in any structural or numerical changes in chromosome is known as chromosomal aberration. It is basically of two types. One is structural and another is numerical. When the variations is found in the structural part, like changing in structures of the chromosome, it is called as structural aberration. And when the change in numerical positions of base pairs or genes changes, it is called as structural aberration. Variation in the structure and number of chromosomes have been observed in natural population, and it could also be produced artificially in a variety of organisms. So then discuss about the structural changes in chromosome. What is the structural changes in chromosome? Any visual changes in the structure of chromosome or any kind of abnormalities in the chromosome is known as structural changes in chromosome. So it can be occurs by four different processes. First one is deletion, then duplication, then inversion, and then translocation. Let's discuss each and every processes in details. So the first is deletion. What is deletion? The meaning of deletion is lose a part, like losing a specific segment from the particular normal complement of gene. We can also call this as deficiency because it lose a part. The smaller deficiency present in heterozygous condition can be tolerated by an organism. The most of the deletion can lead to death of an organism. Deletion have been observed in Drosophila, maize, tomato, etc. Then, what are the different types of deletion? Basically, there are two types of deletion. One is terminal and another is intercalary. What is terminal deletion? When the deletion take part on the top or the terminal position of the arm of the chromosome, it is called as terminal deletion. The example are 4q terminal deletion syndrome, 6q terminal deletion syndrome, 1p36 deletion syndrome, etc. Then intercalary deletion. When the intermediate portion of a chromosome get deleted, then it is called as intercalary deletion. Here the deletion takes place on the arm, not at the terminal position but at the intercalary position. For this, two breaks are needed. When two breaks occurs, the segment is detached from the arm and get deleted. Example: CRI chat syndrome, 4p deletion. Then pseudodominance. At P-chitin stage in bivalent conditions, the chromosomes are fine to form a loop. In presence of a deletion, a recessive allele will behaves as a dominant allele. This is called as pseudodominance. Means when a dominant or recessive part of a chromosome get deleted, then the recessive allele behaves as the dominant allele and express itself. then it is called as pseudodominance. The recessive allele is not dominant but act as dominant in the presence of deletion. So it is called as pseudodominance. It was observed in Drosophila and maize by L.G. Stader. He firstly proposed that during induced radiation, the recessive allele will act as dominant allele and shows pseudodominance. So, first time LG studied what observed it and proposed it. Then this is the mechanism how it occurs. When ABC, small a, small b, small c gene is crossed with capital A, capital B, capital C gene, then the small a, small b, small c is the recessive and the capital A, capital B, capital C is the dominant part. But due to radiation, the deletion occurs in the capital B region. So the gametes form will be from recessive, it will be small a, small b, small c, and in dominant it will be capital A, capital B, capital C, and capital A and capital C. When it crossed, then in the F1 generation, in the next generation, we get capital A, capital B, capital C, and ABC. And ABC and AC, which is not completely expressed, but small ABC is expressed itself, so it is called as pseudodominance. First, a loop formation occurs, then the process starts. It occurs during the P-chitin stage. So let's study the theory part. But when ABC is IR radiated, then deletion may used to leading to the expression of pseudodominance. The P-chitin stage of the meiosis is examined. The presence of loop will indicates the location of gene. During deletion, we can locate the position of gene by studying the loop during the P-chitin stage. Then duplication. This is the addition of a part of chromosome that exists on the same chromosome. So basically यहाँ पे होता क्या है, एक सेगमेंट है जीन का, क्रोमोसोम में एक सेगमेंट है और वही सेगमेंट का एक डुप्लीकेट पार्ट उसी क्रोमोसोम में प्रेजेंट होगा. सो इंक्रीज करेगा, यहाँ पे साइज में इंक्रीज होगा and it is called as duplication. Based on the location of duplication, it is of 4 types. That is, in the adjacent region of homologous chromosome, at displaced position of the chromosome, on different arm of the same chromosome, on different chromosome. So let's understand by some images. The first one is duplication at adjacent region. So in the first strand, that is 1 2 3 4 1 2 3 4 in both the strand, and in the second generation it will gives the 1 2 3 4 in first, but 3 4 and 3 4 get duplicated. This is the duplication occurs at adjacent region. वो दोनों पास पास में duplicate हो रहे हैं. Then at displaced position of chromosome. In the second image, 2 3 get duplicated, but they are not present in adjacent position. In between 2 3 and 2 3, 4 is present. So this is at displaced position. Then on different arm of same chromosome, it takes place in two different arms of the same chromosome, 2323 2323. Then on different chromosome, here 2 3 get duplicated on the different chromosome, 2 3 placed in the different chromosome, so it is called as on different chromosome. Then one of the example in Drosophila is the formation of barred eye. The barred eye means eye are narrow as compared to normal eyes. This phenotypic character is due to the duplication of a part of chromosomes. In Drosophila, it can be demonstrated that the barred eye character was due to the duplication in the 16A region of X chromosome. Then, inversion. It involves a reverse order of the gene in a part of chromosome. So basically यहाँ पे एक सेगमेंट क्या हो जाता है, रिवर्स हो जाता है उसके सीक्वेंस में, वो रोटेट कर जाएगा. An inverse produced where there are two breaks in the chromosome. दो breaks आएंगे. Now the intercalary segment rotate in 180 degree in a reverse order. And after the two break, the segment that rotate in 180 degree in a reverse order. So it is of again two types. One is paracentric inversion and pericentric inversion. So paracentric inversion. This inversions where the inverted segment does not include the centromere. सेंट्रोमियर वाले पार्ट पे कोई भी ब्रेकेज नहीं होगा और कोई रोटेशन नहीं होगा. एक सिंगल आर्म पे टू ब्रेक्स आएंगे और जो सेगमेंट बनेगा वो सेगमेंट रोटेट कर जाएगा और फिर से रीजॉइनिंग हो जाएगी. देन इट इज कॉल्ड एज paracentric inversion. This is basically on a single arm. Then pericentric inversion. Here the centromere is totally involved. The inverted segment include the centromere. Sometimes it is responsible for the evolution. Example, salivary gland of Drosophila. The 17th chromosome in chimpanzee is metacentric, which evolved through the pericentric evolution and forms apocentric chromosome in 17th human chromosome. It also occurs in plants. Then translocation. A chromosomal translocation is a type of chromosomal abnormality where a piece of one chromosome breaks off and attached to the another chromosome. This can result in changes in gene order and potentially leads to genetic disorders or increased risk of cancer. So types of translocation. One is reciprocal translocation. Two chromosomes exchange segments with each other. Then Robertsonian translocation. An entire chromosome attached to another at the centromere. So reciprocal में क्या होगा? Two chromosome exchange their segment with each other. Here एक basically एक segment exchange होगा क्रोमोसोम के बीच में, बट इन रॉबर्ट्सोनियन में क्या होगा कि डायरेक्ट सेंट्रोमियर के पोजीशन से डिटैच्ड होके एंटायर क्रोमोसोम अटैच हो जाएगा दूसरे के साथ विथ सेंट्रोमियर. सो दैट्स द इमेजेस representing both the translocation, and that's all for the today, and thank you for listening.